Hi everyone, in this video we're going to continue with our previous nucleic acids lecture, specifically focusing on the structures of sugars that are involved in individual nucleotides, as well as the general nucleic acid structure, especially in the context of forming a double helix DNA molecule. Now, in this slide, we're going to look specifically into the sugar component because it's very important in that it's one of the most direct chemical differences between an RNA and a DNA molecule. Now, a DNA molecule is going to contain a sugar that is known as deoxyribose, whilst in an RNA molecule, we're going to have the sugar called ribose. And the formulas are given here, wherein for deoxyribose, where we have C5H10 and O4. For ribose, we have C5H10 and O5 as opposed to O4. So basically, ribose sugar has one additional oxygen compared to a deoxyribose sugar. And this is kind of really literally mentioned in the name deoxyribose, the absence of oxygen, which has less oxygen in comparison to ribose. And the reason why this is so important is because it directly relates to the chemical stability or how reactive each of DNA or RNA molecules are going to be. Basically, by having this additional oxygen, ribose is going to have a hydroxyl group right here, as opposed to deoxyribose, which just has a hydrogen group kind of on its own. And this additional hydroxyl group makes this RNA molecule much more reactive than the DNA molecule. Hence why we have the RNA world hypothesis, where we kind of just hypothesize that ribose can, in addition to storing and passing on genetic information, function as a catalytic enzyme called ribozyme. But again, that's a story for another video. And in this pentose molecule monosaccharides, both of them are pentose monosaccharides, they're going to have 5 carbon atoms, and they commonly appear in the sort of pentagonal ring structure, as you can see here. Right, next we're going to move on to the structure and how these individual nucleotides are linked together in order to form a single nucleic acid strand. Now, each nucleotide is connected through the interaction between its sugar and a phosphate group. The phosphate group is bonded to the 5th carbon, also known as 5' carbon of the pentose sugar, and this end of the nucleotide is altogether referred to as the 5' end. Now, on the opposite end, we have a 3rd carbon, also known as 3' carbon, and this carbon forms a bond with the phosphate group of the next nucleotide in the chain. Right, so we can kind of see that this bond is forming right here, whereas we have this bond forming here between the phosphate group, and the 5 carbon group. Now, as the nucleotides kind of link in this manner, they form a repeating chain of alternating sugars and phosphates, and this is known as the sugar-phosphate backbone. And these linkages are formed through what's called a condensation reaction. Kind of to freshen things up, condensation reaction is just removing water, 1 molecule of water, as these bonds form, and it's the opposite of the hydrolysis reaction, which is adding water to break bonds. These are a classic case of anabolic and catabolic reactions. Anabolic meaning that we're building up larger molecules from smaller units being linked and bonded together. Now, the sugar-phosphate backbone, the specific bond that's involved in linking these nucleotides together is the phosphodiester bond. And this bond essentially just forms between the phosphate group that's attached to the 5' carbon of one sugar and 3' carbon of the adjacent sugar. So while the sugar and the phosphate form the backbones, we do have the nitrogenous bases, and it's attached kind of separately. Each base is bonded to the 1st carbon, or 1' carbon of the sugar. Now, this particular arrangement where we have this backbone remaining consistent along the strand and the base projecting outwards, it's really important because the base projecting outwards can kind of readily interact with the complementary bases on the other strand in the context of forming a double helix in a DNA molecule. Now, DNA has a very unique, distinctive double helix structure, meaning that it is composed of 2 nucleotide strands that are twisted or intertwined around each other. And the 2 strands are described as anti-parallel, meaning that they run parallel to each other, but in opposite directions. So one strand is going to run from the 5' to 3' end, whilst the other runs from a 3' to 5' sort of direction. Now, despite running in opposite directions, both of the strands do follow the rule that nucleic acids are generally synthesized from the 5' to 3' direction. And these 2 strands are held together by hydrogen bonds between the complementary nitrogenous bases. And as we know, adenine is going to bind with thymine, while guanine binds with cytosine. And of course, we do remember the number of hydrogen bonds here, where between the AT base pairs, we have 2 hydrogen bonds. Between the CG, we have 3.